Good afternoon, everyone. Today's class is on specialized connective tissue, cartilage. But before we go on to cartilage, let's recap what we've done so far. You've talked about epithelium and we've studied about epithelium. The epithelium consists of cells which are lying in close apposition with each other with very little extracellular matrix or spaces in between them. They lie on a basement membrane and they line either the external body surfaces or the internal body cavities. The basement membrane separates this epithelium from the connective tissue. The connective tissue also consists of cells. This is a fibroblast, mast cell, plasma cell, fat cell, macrophages. So this also consists of cells. The cells may be resident cells or cells which have migrated from somewhere else. But the cells here are separated from each other by extracellular matrix, which consists of fibers and ground substance. The fibers could be the reticular fibers, elastic fibers, or collagen fibers. And the ground substance has a gel-like consistency and is made up of glycosaminoglycans and proteoglycans. Now, depending upon the types of cells which are present, the number of fibers, what is the characteristic features of the ground substance, the connective tissue is classified further. Now, depending upon the ratio of the cells to the fibers and the amount of cells, the number of cells, the amount of fibers or the ground substance which is present, the connective tissue has been further classified in connective tissue proper, which consists of the loose connective tissue when the number of cells are more and the fibers are less, like the areola, the adipose, and the reticular connective tissue. And it is also defined as dense connective tissue when the number of fibers are more as compared. The ratio of the fibers to cell, the fibers are more. This was dense regular connective tissue like the tendons and dense irregular connective tissue. Now this you've already covered in your lecture as well as practical. The focus of the next few classes will be to talk about the specialized connective tissue or the connective tissue of the skeletal system. And in this class, that is the cartilage and bone. And in this class, we will be focusing on cartilage. Now, these connective tissues differ from the other, from the connective tissue proper because of the properties, the type of cells which are present, the properties of the ground substance, and the kind of fibers which are present in it. There is another specialized group of connective tissue, blood, which you basically cover in physiology. The competency which we will be covering is identify cartilage under the microscope and describe various types and structure function correlation of the same. The learning objectives are identify and describe the 3 different types of cartilage, compare the organization and content of cells, fibers, ground substance within different types of cartilage, describe the maturation process of these cartilages. The cartilage is a solid, weight-bearing connective tissue which lacks the rigidity of the bone. It is strong but deformable. It is a pliant, load-bearing connective tissue. In prenatal life, you are aware that most of the skeleton is made up of cartilage, but postnatally, it is present at certain sites. It is present within the skeletal system where it forms the articular cartilage, which is present between the joints. It forms the menisci, the fibrocartilage menisci, which you see in between the joints. It also forms the intervertebral disc, which is present between the 2 vertebrae. It forms a part. Forms a part of the thoracic skeleton in the form of costal cartilages. It also forms the skeletal framework of the auricle, which is an elastic cartilage. It forms the skeletal framework of the nasal cavity, the larynx, trachea, as well as the larger respiratory tubes. Wherever there is cartilage, the cartilage is there wherever we need to bear mechanical stress and have shock absorbing capacity because it provides cushioning effect to the joints. It is strong but deformable. It supports the soft tissue of the nose, nasal cavity, of the ear, and it is essential for development and growth of long bones because you know the growth plate which is present is also nothing but hyaline cartilage. Like the other connective tissues, the cartilage consists of cells These are the specialized cells of cartilage known as chondrocytes. And you can see that these chondrocytes are not lying in close apposition to each other, but are separated from each other by this pinkish substance which is present out here, which is the ground substance, which is basically the extracellular matrix, which consists of the ground substance as well as fibers. Now, what is the ground substance? What are the fibers which are present? What are the properties of the chondrocytes? Let's study about, and how do they help in the function? Let's study about this first and then go on to the different types of cartilages. Before we go on to see the histological appearance of the cartilage, let's see what is the organization of the various components of a cartilage. Now, suppose we take a prototype, say a hyaline cartilage, which is we are taking from the trachea. So, you know, the trachea looks like this in the body. It's dividing into the bronchi, and each of this is made up of a tracheal ring. This entire skeleton is made up of tracheal ring. So, if we were to take this ring, it would be a C-shaped ring which is bounded by smooth muscle on the posterior aspect. From this, we pick up this particular part of it. All right. So, if we were to draw this on the outside, that is out here, it would have a layer of perichondrium, which is made up of type 1 collagen fibers. So, this is the perichondrium, which is nothing but dense fibrous connective tissue which separates the cartilage from the surrounding structures. This is made up of type 1 collagen fibers. And when we look at these type 1 collagen fibers under the light microscope, they appear wavy and crimped. And that is why we've drawn it like this. Even on this side, on the other side, we would have similar perichondrium made up of type 1 collagen fibers. Adjacent to it, part of the perichondrium are also going to be young cells or the chondroblasts, which we would see. And they would form the cellular layer of the perichondrium. We'll just make the cells properly. This is not a histological diagram. This is just to give you an idea of the structure. In between would be the chondrocytes and the extracellular matrix. So if we had these, this would be the cells of the cartilage. Again, I'm not drawing the correct histological structure of it right now. I will come to it. This is just a 3D diagram to make you understand, and then we will draw the diagram. So there are these groups of cells or singly placed cells which are going to be present. Now in between the cells should be fibers as well as the matrix. So what fibers are present? Number one, we will have type 2 collagen fibrils forming a 3D lattice network. So even though they appear as if they are randomly placed, there is an orientation to it which is present. These are type 2 collagen fibrils. Now, fibrils are smaller than the fibers. That was fibers. This is fibrils. They are about less than 50 nanometers in diameter, and they are not picked up by the light microscope. So they are usually not visible in our histological slides. So if you remember the slide which we just saw earlier, we could see the cells and we saw this pinkish hue structure which was there, which I said was the extracellular matrix. But you did not see any fibers running through it because these collagen fibrils are very small, which is beyond the resolution of a light microscope. They are about less than 50 nanometers in diameter. And they form a nice, well-oriented 3D network. Now, in between these fibers, and in case it's an elastic cartilage, we will have elastic fibers also which are present. And in case it is the fibrocartilage, which we'll discuss later, there will be type 1 collagen fibers running within the substance. So depending upon the type of cartilage, the fibers will vary, but this will always be there. The collagen fibrils will be there, but we may have an addition of elastic fibers or type 1 within the substance. Now, the other structure which is present here is the proteoglycan. This proteoglycan is giving it the semi-rigid consistency which we see in the cartilage. It is forming the ground substance. Now, this proteoglycans consists of proteoglycan aggregates. Is formed in the form of aggregates, and each aggregate consists of one large molecule. अब हम एक aggregate पहले describe करें. It consists of one large molecule of a large carbohydrate molecule made up of disaccharide units, which is known as hyaluronic. Hyaluronic. Non or hyaluronic acid. अपने में जब आप बायोकेमिस्ट्री पढ़ोगे तो आपको पता चलेगा ये कुछ नहीं है, ये ग्लाइकोजेमिनोग्लाइकन है. That is a carbohydrate moiety which is made up of multiple disaccharide units. ठीक है? So let's say this is one hyaluronan molecule and this is one more hyaluron. ऐसे multiple molecules यहाँ पर spread है. Again in a very organized three-dimensional network. Attached to it are present proteoglycan aggregates, which consists of one core protein. This is the core protein. And to this core protein are attached more glycosaminoglycans. And these glycosaminoglycans are chondroitin sulfate, and keratin sulfate. So, ये एक कोर प्रोटीन है. इस कोर प्रोटीन पर ये ग्लाइकोजेमिनोग्लाइकन्स अटैच्ड है, जो या तो कॉन्ड्राइटिन सल्फेट है या केराटिन सल्फेट है. एंड लिंकर प्रोटीन्स के थ्रू ये जो लिंकर प्रोटीन्स ये बने, बाय मींस ऑफ दिस लिंकर प्रोटीन they are attached to this hyaluronan molecule. We have large number of these proteoglycan monomers attached to this particular hyaluronan molecule, giving it a bottle brush appearance. Right? Plus, there are other multi-adhesive glycoproteins and other kinds of proteoglycans also present. But this is the major proteoglycan aggregate which is present, and each of these which is present within the substance of the cartilage. So ऐसे इस वाला जो ये वाला जो hyaluronan molecule है, उसके ऊपर भी ऐसे ही ये linker proteins के through ये core proteoglycan monomers attached होंगे. Now, as a result of it, you have so much of negative charge which is present within the substance because of the sulfate groups which are present with the chondroitin sulfate and keratin sulfate, as well as the carboxyl groups. These interact and entrap the water molecules. So the molecules are trapped and entrapped by these glycosaminoglycans and the proteoglycan monomers to form a 3D network and to form a semi-rigid consistent structure. ये जो collagen fibrils है और जो ये hyaluronan molecules है, ये भी एक दूसरे के साथ electrostatically interact कर रहे हैं. Now, because so much of water is trapped, 60 to 80% of the cartilage weight is nothing but water. Approximately 60 to 80% of the cartilage is made up of intercellular water. The net weight of the cartilage is basically water. And as a result of it, it has this compressive turgor which we talked about, the ability to take up the mechanical stresses, to absorb the shock, because एक gel-like semi-rigid, एक semi-rigid structure एक बन पाता है because of this entire structure or the internal structure of the cartilage. And a lot of this histological structures which we will be seeing will also be because of this water being lost during processing. So this is important to understand how the cartilage is made up of. What is the 3D structure of a cartilage when we look at it? So क्या-क्या पढ़ा हुआ है? अब जब भी हम cartilage के बारे में बात करेंगे, we should be able to talk about the cells of the cartilage, which could be chondroblasts or chondrocytes. We should be able to talk about the collagen fibers which are present, which are various types of collagen fibers, mainly type 1 and type 2 for a UG student. There are more for the others under postgraduate students, which they will study on their own. Plus, we have proteoglycan. We also have a large number of proteoglycan, which is present in the form of aggregates, which is made up of proteoglycan monomers, which in turn are made up of proteins and glycosaminoglycans. So ये पूरा का पूरा structure आपको आना चाहिए, and consequent to the sulfate which is present in the sulfate and the carboxyl molecules of the glycosaminoglycans, a large amount of water gets trapped by the cartilage. So this is how the cartilage fibers, the cartilage ground substance and the chondrocytes or the chondroblasts are interacting with each other. और ये सब किसने बनाया? ये जो gags है, ये जो collagen fibrils है, ये collagen molecule किसने बनाया? ये सब chondroblasts and chondrocytes ने बनाया. So they are the ones who are actually responsible for making this. So they should be active protein secreting cells. And loss of this proteoglycan content or decrease, and as would result in decrease in the water content, and that would result in diseases, would result in degenerative diseases of the joints like the osteoarthritis, which you see the joint pain which your grandparents sometimes complain of is because of the change in the milieu, this dynamic structure of the cartilage, the changes in this dynamic structure of the cartilage, the changes in the chondrocytes, the changes in the proteoglycan content would bring about old age changes of osteoarthritis or joint diseases. The resident cells of the cartilage are the chondroblasts or the chondrocytes. The chondroblasts are the young active cells, while the chondrocytes may be active, that is the younger ones, or older ones. So these are the young dividing cells, while the chondrocytes may be active chondrocytes, which are secreting the matrix, making the proteins, dividing, or the older chondrocytes. Now let's start with the structure of these. The chondroblasts are flattened, irregular cells, elliptical cells with an elliptical nuclei. So the young cells are the flattened, elliptical cells. They are active. They have all the machinery which is needed for protein synthesis with an elliptical or an elongate nuclei. They are best observed near the perichondrium. Below the perichondrium, if you remember, we had made these young cells. So they are best observed there. Then come on to the chondrocytes. The chondrocytes, so what has happened? These chondroblasts, when they become older, when they get surrounded by the matrix which they are secreting, which they are only secreting, they form chondrocytes, right? An active chondrocyte would be oval or irregular in shape, a larger cell with a euchromatic or an open nucleus. That tells you that the cell is actively involved in protein synthesis. Now, they will be present. Okay, we just come back to this. Now, this is how it would look in a light microscope. But if we were to use an electron microscope to visualize it, in that case, we would see that this would be the cell. This would be, here would be the nucleus. When we're looking at the electron microscope, which is an active nucleus, it would be having, there would be these rough endoplasmic reticulum with ribosomes. The Golgi apparatus would be there to package the proteins which it's secreting. So this RERs, the rough endoplasmic reticulum with ribosome, would give the cytoplasm a basophilic hue. and there would be the Golgi apparatus which would also be present. Plus, we would see some glycogen droplets. A more mature cell or an older cell would appear again as a large cell with a heterochromatin, that is a closed, dark colored, bluish heterochromatin nucleus, right? With empty spaces in it. Because this, or again, with electron microscope, we would have seen that this cell has large number of these glycogen droplets as well as lipid droplets which are present out here. जैसे जैसे older होता जा रहा है, उसके अंदर लिपिड ड्रॉपलेट्स और ग्लाइकोजन ग्रैन्यूल्स ज्यादा बढ़ते जा रहे हैं, उसकी मशीनरी लेस एक्टिव होती जा रही है, उसके पास होंगे रफ एंडोप्लाज्मिक रेटिकुलम, but it's now going into a resting kind of a phase. Now, why is this important? Because when we look at it under the microscope, जब हम histological processing करते हैं, because of these lipid droplets, these lipid deposits as well as the glycogen droplets, during processing they are lost and the cell tends to shrink. So what we see under the light microscope is a cell which is surrounded by an irregularly shaped cell with its nucleus, and there is this empty space around it which we call the lacuna. So actually क्या था, this entire cell, ये जो हमारा cell है, this cell was occupying this entire space, और फिर उसके around उसने यहाँ पर ऐसे 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 अपना extracellular matrix secrete करा. But जब हमने उसको histologically process किया, जब हमने इसको various grades of alcohol, water से pass किया, तब क्या हुआ, the lipid droplets as well as the glycogen droplets got lost. So now the cell shrunk and we can see this empty space around it which we call as the lacuna, which is where actually the cell was sitting. The cell in an intact cartilage was occupying this entire space. It is an artifact. This lacuna is an artifact, and we say this is where it had been sitting. Now, these cells, which we are going to see, these chondrocytes, may be singly placed, like we are seeing out here, or in some cartilages, as we'll see, they will be forming these isogenic groups, of chondrocytes. That means this individual cell has divided to give rise to these daughter cells. And these daughter cells are still laying down their matrix. As they keep laying down the matrix, the extracellular matrix. Remember, that was the function of these chondrocytes. It was the function of these chondrocytes to secrete the matrix and the fibers. As they start secreting the matrix, they will start separating from each other. But an isogenous cell group tells you that they represent the cells that have recently divided. So in the histological slides, you will see the chondroblasts. So now look at this. This is the perichondrium. Right, we can identify this is the perichondrium. What are we seeing? Type 1 collagen fibers forming dense connective tissue on the periphery. Then below that, we would look for the chondroblasts, which lie close to the surface. These are flattened elliptical cells with an elongate nucleus. Right. As they secrete extracellular matrix and get surrounded by it, they result in the formation of these chondrocytes. Now look very closely. Look at this particular. Can you see the cytoplasm around here? This pinkish hue of cytoplasm? Or look at this one, the pinkish hue of cytoplasm with the nucleus which is present out here, a darkened nucleus. Probably you can see a dark nucleus out here. And look at this white space surrounding this. This is the lacuna and this here is the chondrocyte. The chondrocytes lie within the cartilage in the slide in a lacunae. This is the lacunae which we are seeing. They actually secrete all this matrix which is present around it. Now look at these two. They are in close contact with each other. They are in apposition to each other. So that means this one of these cells has divided and given rise to this isogenous cell group. Or look at this isogenous cell group. And as they secrete the matrix, as these individual chondrocytes keep secreting the matrix and surrounding themselves, they will start separating from each other. So this cluster, this group of 2 cells out here, or there may be a group of 4 cells, which we may see, this forms an isogenous group. The perichondrium is the vascularized connective tissue sheath which is surrounding the cartilage and separating it from the surrounding structures which are present. For in this case, you can see there is connective tissue present here, and this is the cartilage, and the perichondrium is separating it from it. The perichondrium consists of an outer fibrous layer, which you see here, and an inner chondrogenic layer. The outer fibrous layer is composed of type 1 collagen. It also contains the blood vessels which are there. Thereafter, and it also contains some cells. These are the cells which are the resident cells of the dense connective tissue, which is the fibroblasts. Thereafter is this inner cellular layer, which is consisting of chondroblasts, which gives rise to more cartilage cells. Now, the perichondrium is absent in case of an articular cartilage as well as in case of a fibrocartilage. So how does the cartilage get its nutrition? So the blood vessels which are present in the perichondrium, from there the nutrients diffuse through this gel-like matrix, extracellular matrix, which is made towards the chondrocytes. So that is the importance of this semi-rigid gel-like, this water content of which was present in the extracellular matrix, that it helped the avascular cartilage to get its nutrition by diffusion, and the waste products similarly are sent away through this matrix into the blood vessels which are present in the perichondrium. So the cartilage is avascular. That is important. It has clinical importance to it. The cartilage is avascular, number 1. It gets its nutrition by diffusion. And number 2, the importance of perichondrium is that it is needed for growth and maintenance of the cartilage. So perichondrium is responsible for the nutrition because it has the blood vessels in it, and it is needed for the growth and maintenance of the cartilage because it has the chondroblasts which are present in it, and it also has fibroblasts which can get converted into the chondroblasts if the need arises. On the basis of the predominant fibers which are present in the cartilage matrix, the cartilage is divided into 3 types: hyaline cartilage, elastic cartilage, and fibrocartilage. In hyaline cartilage, the predominant fiber type is type 2 collagen fibrils. In elastic, apart from the type 2 collagen fibrils, the elastic fibers are present. Whereas in type 3, we have both type 1, whereas in the fibrocartilage, we have both type 1 and type 2 collagen fibers present. Wherever we want to resist compression, provide cushioning effect, wherever you want structural support, that is the place where hyaline cartilage is going to be present. So it is it makes up the costal cartilages, it makes up the articular cartilage which is present, it forms the epiphyseal plates, it forms the skeletal framework of the parts of some of the larynx, for example, the thyroid cartilage, the cricoid cartilage, the nasal bones, they are all the nasal cartilage are all hyaline in nature. Whereas the elastic cartilage is present wherever we want to provide a flexible support so that we can pull your ears. So the pinna, the external ear, the external acoustic meatus, the eustachian tubes, the epiglottis of the larynx, all of them are made up of elastic cartilage. So E say elastic. So what was that? The external ear, external acoustic meatus, eustachian tube, epiglottis. And the other small cartilages of the larynx are all made up of elastic cartilage. In contrast, the fibrocartilage is present. It forms the intervertebral disc. wherever you want to resist deformation under stress. So where is it present? It is present where you want to have the shock absorbing capacity. So it is present in the menisci, you saw in the knee joint. It is present in the intervertebral discs, which we see. We've also seen it in the sternoclavicular joint as the articular disc. It was the fibrocartilage. So these are the three different types of cartilage. And now let's study the histological structure of each one of them as to how they will appear under the microscope. This is a slide of the hyaline cartilage. The hyaline cartilage consists of an outer layer of perichondrium. This is the perichondrium, which consists of a dense fibrous layer, which is made up of collagen, and an inner cellular layer. This is the inner cellular layer. The perichondrium is separating it from the surrounding connective tissue. The perichondrium is on both sides. In between here is the mass of the cartilage which is present, which consists of cells which are either placed in groups or singly placed. These cells are the chondrocytes, and in between them is the matrix of the cartilage. Now, let's see the matrix first. Now, the matrix, as we had studied earlier, should have collagen fibrils, and it should have the various glycosaminoglycans and the proteoglycans. But all that is not seen. What we can see is that number one, that there are no fibers seen. Because if you remember, the bulk of the structure of the hyaline cartilage was formed by type 2 collagen fibrils. And these collagen fibrils had submicroscopic dimensions. That is, they were less than 20 nanometers in diameter, and they were beyond the resolution of the light microscope. Number two, their refractive index is similar to the refractive index of the ground substance, and hence they are not visible to us when we stain a slide with HNE and observe it under the light microscope. Now, the other thing which you can see is that the matrix appears bluish in color. Now, this bluish tinge to the matrix, or the hematoxylin staining of the matrix, is seen only if the cartilage tissue is obtained fresh. It is kept cool during fixation. Because at that particular point in time, if we maintain these conditions, the sulfated glycosaminoglycans can be preserved. And if they are preserved, the negatively charged sulfate molecules of this glycosaminoglycans attract the hematoxylin dye and bring about a basophilic hue. But mostly in our slides, we will see it as pinkish in color like this. But still, we are going to say that the matrix is homogeneous, because we cannot see the fibers in it. That is, it's homogeneous, it is translucent, and it has a bluish tinge, it shows basophilia or has a bluish tinge to it, right? Now, look at the individual cells. Now, let's come on to this particular part of it. Now, the cells may be, when we are looking at it at higher resolution, the cells may be singularly placed, or they may be in isogenous cell clusters. If we see an isogenous cell cluster, we know that these cells are a result of mitotic division of one of the cells, and as a result, they have resulted in the formation of a cell cluster. Now look at the singly placed cell. Can you see? This is the nucleus. Oops. This here is the nucleus. This is the bluish cytoplasm which you're seeing, and this would be the lacunae. So this is the cytoplasm of the cell, and this is the nucleus which you're seeing in the singly placed cell. Do you remember? The lacunae, which is here, this whitish space, is a shrinkage artifact which appeared. Why did it appear? It appeared because the cell shrunk, and we can see where it had been lying in the matrix. This is the surrounding matrix. Now, even if you look closely at the matrix, although I said it was homogeneous, because here I was referring to the fact that there are no fibers which are seen within the matrix whatsoever. It's just one glassy translucent structure which you're seeing. But if you observe carefully, you can see a difference in staining characteristic. There are certain regions which are staining darker, there as compared to the other areas which are staining comparatively lighter. So the matrix also has its own characteristic features of staining. Now, the staining characteristics of matrix differ from one another because of the concentration of sulfated glycosaminoglycans. Now, just in the right into the vicinity, near the chondrocyte. Now look at this chondrocyte, right in its vicinity. Do you see where the blue mark arrow is pointing? This part of the matrix is known as the capsular matrix. This is most intense in staining because it has the highest concentration of the sulfated glycosaminoglycans which are seen. So it forms like a capsule around the cell. So this blue arrow is pointing towards this thing which is right around the cell. So ये उसका capsular matrix है, which is highly sulfate. It consists of highly large amount of sulfated glycosaminoglycans which have been just synthesized by the cell and are in the vicinity of the cell. So this is known as the capsular or pericellular matrix. ये lacunae है, ये न्यूक्लियस है और ये उसका सेल का साइटोप्लाज्म होगा. ठीक है. Now furthermore, if you move further across this area, this has lower concentration of sulfated glycosaminoglycans as compared to the capsular or the pericellular matrix, and this area is known as territorial matrix. This is surrounding a group of isogenous cell clusters in this particular case, but even in a singly placed cell, you may see a darker region around the cell which is known as the territorial matrix. And even further, in between here, where the concentration of glycosaminoglycans is the lowest, this area is known as the interterritorial matrix. So the matrix has different staining characteristics, and this is because of the different concentration of sulfated glycosaminoglycans which are present. Right in the vicinity, it is known as the capsular matrix because it's surrounding the cell like a capsule or a pericellular matrix. Then comes the territorial matrix, and in between is the interterritorial matrix. Now, another thing to note out here is that the cells are either singly placed or forming an isogenous cell cluster. No fibers are seen, so it is homogeneous, but it is not homogeneous in staining. And out here, right next to the perichondrium, we can see the young chondrocytes and the chondroblasts. These would be the fibroblast cells of the perichondrium, and these would be the young chondrocytes which we are seeing, which are flat and elliptical in shape, which are present near the perichondrium. So once again, the important points to remember are that the hyaline cartilage contains type 2 fibrils that have the same refractive index as the ground substance, and also they have submicroscopic dimensions less than 50 nanometers in diameter. Therefore, they are not visible with light microscope. The matrix stains blue with conventional dyes due to the relative abundance of sulfate groups of the proteoglycans or the glycosaminoglycans in its ground substance. And the cartilage possesses numerous isogenous groups. Now, if you were to make an isogenous group, please see how you're going to make. If you make a singly placed chondrocyte, so this would be the capsular matrix, then this would be the cell. This would be the chondrocyte. You would stain it lightly with the eosin color and put in a nucleus out here. This is how it would look when we are making a single chondrocyte. And when we make an isogenous cell group, there are these cell clusters. 1 minute. There is going to be some matrix in between. Please remember, that's why. So, ये आप cartilage cell होगा, जो वो lacunae में पड़ा हुआ है, and this would be its nucleus. This is an isogenous cell cluster. So there is, this is the lacuna. This would be the matrix. This would be the cell with its nucleus. So when you make it, obviously you will color it with eosin. If I can't do that. So please remember, when you make this diagram in the files, please make it carefully. Even the single place cell may be like this also. So ये उसका lacuna है. ये यहाँ पर lacuna है. और ये क्या है? Chondrocytes का. This is how you will make your chondrocytes. The elastic cartilage occurs in the external ear, epiglottis, external auditory canal, eustachian tube, and parts of the larynx, the small cartilages of the larynx, that is the cuneiform and the corniculate cartilages. They are present wherever we need the property of elasticity along with flexibility and support is required. The elastic cartilage has all the characteristics of hyaline cartilage in the matrix. Plus, it has interlacing elastic fibers. So the fibers which are going to be present are similar. That is, the collagen fibrils will also be present, which we cannot see. Plus, they would be interlacing elastic fibers. In contrast to the hyaline cartilage, they have fewer isogenous group. Now, let's look at a slide and see how it appears. Now, this is a slide from our own lab. You can see this out here is the hyaline cartilage. The elastic cartilage. पहली चीज है कि ऐसे पता चलेगा कि हम cartilage की slide दिख रहे हैं. So you have to be able to recognize. I told you, बड़े बड़े cells दिख रहे होंगे chondrocytes, उल्लू के आंखों के जैसे. So a large number of cells will be seen in an area. So you know that you're looking at us with some matrix in between. तो आपको पता चलना चाहिए कि मैं एक slide दिख रहा हूँ जो एक cartilage की है. Now to differentiate, we will differentiate a hyaline cartilage from an elastic cartilage. Now in the slide of an elastic cartilage, which is seen here, there is again a perichondrium on the outside, right? There is a perichondrium on the outside, and this is the cartilage which we are seeing. You can see that the cells are mostly singly placed. Instead of having isogenous groups, you see singly placed cells. So this is a chondrocyte. This is the lacuna, and this out here would be the chondrocyte. And within the chondrocyte, here you can see even a nuclei which is present. Look at here also. But there is a nuclei in this particular cell. You can see the nuclei, you can see the lacuna, and you can see the chondrocyte. So they are singly placed cells. And again, no fibers are seen, because elastic fibers are seen in a slide only when we stain them with special stains. In case of cartilage, when we use special stains like Orcine, we would be able to see the elastic fibers present here. At a higher mag, again look at the same thing. Look at the cartilage cells. You can see the cells very well. The cells have shrunk. Why have the cells shrunk? Because the cells have lipid deposits and glycogen droplets in it. And during processing, when they are lost, the cells tend to shrink. Now, another thing which should strike you when you're comparing a hyaline cartilage from an elastic cartilage is that the ratio of the cells per unit area is more in case of a slide of an elastic cartilage. And that also helps us to differentiate between an elastic cartilage and an hyaline cartilage cell. And a hyaline cartilage slide. So this is when we would have stained it with a special dye that is the Orcine. That is when the elastic fibers are seen. These are the singly placed cells. Again, there is perichondrium on the outside. This perichondrium on the outside, and this is the matrix of the cartilage in which special fibers, that is the elastic fibers, are seen. Rest of the properties, the chondrocytes, the chondroblasts, the matrix properties, that the glycosaminoglycans, they're all similar in case of the hyaline cartilage as well as the elastic cartilage. The fibrocartilage. Now, the fibrocartilage is a hybrid between dense connective tissue and hyaline cartilage. Dense, regular connective tissue and hyaline cartilage. So here, wherever you need compression, great tensile strength with flexibility, it is found where a tendon or a ligament attaches to a bone, and it is located in the intervertebral discs, in the pubic symphysis, as well as in the menisci, which we saw in the knee. Now, how would a slide appear here? Now you can see that the characteristic feature here should be the ground substance is less, number one, and the fibers are more. And now the fibers all of a sudden are visible to you. They are wavy, they are crimpled. So it's type 1 collagen fibers. So the slide of a fibrocartilage or a fibrocartilage consists of type 1 collagen fibers plus collagen fibrils, which are not visible. And the proportion of the collagen fibers to fibrils varies with the site of the fibrocartilage where we are seeing. And present within the slide are these cells. देखिए यहाँ भी हमको cells दिख रहे हैं, ये भी हमको cell दिख रहा है और एक ये भी दिख रहा है. So ये जो flattened appearing cells है, these are the fibroblasts. which are the constituents of the dense connective tissue, and this is a hybrid. So we can see fibroblasts, but there are these cells also which have a rounded nuclei. So this cell with the rounded nuclei is the cell of a chondrocyte. So you have fibers, you have bundles and bundles of type 1 collagen fibers which are running. In between, we have cells of chondrocytes which are present, which may be present singly in a row, or sometimes we may also see isogenous cell nests which may be present at places. Plus fibroblasts are also seen. And wherever you see the chondrocytes, only there would be some amount of ground substance which is going to be present. It is important to look for these round nuclei chondrocytes to be able to differentiate between a slide of a tendon, which also had type 1 collagen fibers running in bundles with fibroblasts in between, from the slide of a fibrocartilage. So the fibrocartilage possesses abundant type 1 collagen fibers as well as collagen type 2 fibrils. It has minimal ground substance. The ground substance is usually located in the vicinity of the chondrocytes, which are usually singly placed, but at some times they may be forming an isogenous cell groups also. The round nuclei of the chondrocytes help us to differentiate the chondrocytes from the fibroblasts. Another very important thing to remember with the fibrocartilage is that it lacks perichondrium. The cartilage has the capability of growing. You are aware of the fact that the growth plate is hyaline cartilage, and that growth plate continues to grow. Now, what are the methods by which a cartilage can grow? One of the methods is the interstitial growth, and you've already seen the evidence of that. Now look at this. These are the chondrocytes. Now in this cell, you can see that the chondrocyte is undergoing a process of division, which is mitosis. When cartilage grows from within, that is by mitosis of the cartilaginous cells, to form these isogenous cell nests, this kind of growth is known as interstitial growth. So interstitial growth is when the chondrocytes divides by mitosis and forms these isogenous cell nests of more daughter cells. Initially, the daughter cells share a lacuna, and thereafter, with the secretion of matrix, they tend to disperse from each other. So this mode of growth of cartilage is known as interstitial growth. This is in contrast to the next method of growth, which is the appositional growth. Appositional growth, the fibroblasts which are present in the perichondrium, now they under the influence of certain transcription factors, they become activated, become chondroblasts. This chondroblast starts secreting matrix around it, and then since the matrix keeps getting secreted around it, it goes within, it gets surrounded by more and more matrix, and it forms chondrocyte. So this method of growth is known as appositional growth, that is from above or below, wherever the perichondrium is. And this is appositional growth. So there are two methods of growth of cartilage, one from within, that is interstitial growth, and one from outside, that is appositional growth. Now, there are certain important things to remember with cartilage which are of clinical importance to us. Cartilage can undergo classification with age, especially hyaline cartilage, or only hyaline cartilage actually. In deficiencies in the supply of precursors like minerals, vitamins A, C, and D can lead to abnormal growth and maturation of cartilage. Abnormal production of growth hormone by the anterior pituitary, thyroxine by the thyroid, testosterone and estrogen by gonads, and corticosterone by the adrenal glands can produce also abnormal growth of cartilage. Cartilage can give rise to benign chondromas and malignant chondrosarcomas. So these are the tumors of cartilage. And now, before we end the class, let's do the differences between hyaline cartilage, elastic cartilage, and fibrocartilage. I hope you're able to recognize them when you look at the slides. Hyaline cartilage has an outer layer of perichondrium, so does the elastic, but perichondrium is absent in fibrocartilage. The fiber type present, the dominant fiber type in hyaline cartilage is collagen 2 fibrils. In elastic, apart from collagen 2 fibrils, there are also presence of elastic fibers. In fibrocartilage, we have collagen 1 as well as collagen 2 fibers. The rest of the matrix of sulfated glycosaminoglycans and the protoglycans is mostly common, with some additions in case of certain cartilages. Isogenous cell nests are seen in hyaline cartilage. They are less so common in elastic cartilage and fibrocartilage. The matrix is homogeneous. in appearance. It may have basophilia with it in hyaline cartilage. The number of cells per unit area of matrix is more in case of elastic cartilage, and there is very less matrix in case of fibrocartilage. Now the ground substance is very less. It's basically full of fibers. Ossification is possible in hyaline cartilage. It is not possible in elastic and fibrocartilage. With that, we come to the end of this lecture, and we shall see the slides in the practical.